Hongsheng B will present on impacts of Bering Canyon on zooplankton in the southeastern Bering Sea. Oh, orcas, okay. Talking about zooplankton and having orcas, okay, fair enough. I accept. The floor is yours. Oh, I think I can't hear you. Maybe it's on my end.
No, I think I muted myself, just avoiding making noise. So thank you for the great introduction. And obviously you can see I'm a zooplankton ecologist, but I'm gradually still like not much mesopelagic person. So I'm moving a little bit. I stationed in Chesapeake Biological Lab. So naturally I deal with Chesapeake Bay. This is as far as I can go. The Bering Sea and mostly euphotic zone. But I sample during the night, right? That counts for something without light. So, but anyway, thanks for having me here. And what I'm going to talk about is the impacts of Bering Canyon on the zooplankton distribution. And it's a ongoing project. I'm trying to assemble a lot of different elements together. So we'll see how well I can deliver the proposed project, deliver the message without complete the project. So this is an outline for my talk. It's straightforward. So as we all know, this is a plankton group, right? I don't need to go over this, but because I'm within fisheries group, so I have to study something more related to fisheries. So we know zooplankton is a base for the pelagic food webs. Within the zooplankton group, today I'm going to focus on two organisms. Jellyfish, that's number one. The second is krill. So why I choose those two? Number one, jellyfish is weak swimmer. They literally go with the current. So I can look at how does the shelf, physical process affect their distribution. Number two, why is krill? Because everything in the Bering Sea feed on the krill. So combining with the canyons, I can ask bigger questions. Along the Bering Sea shelf, there are several major canyons. There is features called green belt. So it's a persistent feature. So if we have climate change, the shelf gets warmer, but the shelf break with canyons, they become like a high productivity area. Will they play a role for fish and marine mammals feeding, particularly during the warm years? So the overall things that's what I'm looking at it. So, of course, plankton is important for carbon pump and climate change. They are also excellent indicators for ecosystem-based management. So a little bit about Bering Sea, particularly Southeastern Bering Sea. There are two dominant processes. The Southeastern Bering Sea, particularly the northern part, is ice-driven ecosystems. So during the winter time, they form like ice. Starting from the spring, they melt and release nutrients, promote phytoplankton. However, the time of the melting begins play a bigger role here. If the ice retreat earlier, there's melting earlier, you got late bloom, warm water, mostly small copepods. Of course, we can jump to the next one. When you have small copepods, you got smaller fish, like it's the larger fish, like they don't have enough food, leading to more winter starvations. If the ice retreat late in the spring, then you get earlier bloom and mostly large phytoplankton bloom that promote large copepods. then you get better fisheries, right? That somehow is related to what I'm studying, fisheries. So the second feature is if you look at the Bering Sea, along southeastern Bering Sea, along the shelf, there are lots of canyons, as I mentioned. If you look at this model results for 2015, which is a warm year, see the entire shelf, the green color indicates high productivity. On the shelf, the productivity pretty low, but at the shelf break, that's what you see. So that's sort of like promote the question. You know, we have so many canyons along the shelf break. And from the existing studies, we know that canyons can produce like upwellings. And at the same time, those upwelled water can be transported to on shelf. In other words, if the upwelling elevated productivity, then those productivity could be transported to on shelf through either tidal current or wind-driven current. So there are the mechanisms allow the, say, the canyons affecting the productivities. So the overarching hypothesis in this part is like, can the canyons, areas around the canyons actually serve as refuge areas for upper trophic levels and fish recruitment, particularly during the warm years? So of course, we need to understand the mechanisms and what are the spatial extent for those phenomena. So a little bit about the study site. This is a Bering Canyon area where this like dashed line box surrounded. You can see there are two different colors. One, those in yellow, that's in 2017 and 2018. I was funded by NSF, able to sample like those stations. Each yellow dot is a station. And beside that, what I did is I towed plankton scope from one station while the boat steaming from one station to the next station. For instance, the first line we do is moving this direction. So from one station, I'm towing the system, imaging system up and down throughout the water column for about two hours. That the distance between each station is about 10 to 15 nautical miles, and our towing cover about somewhere like 3 to 4 nautical miles. In other words, we cover a decent amount of area with using the imaging system. You see those three like a very short transect. Those are the surveys we did in 2020, this August 23. So, okay. So because I work on the imaging system, so naturally is some, you know, this is what we do. We did how we sample plankton before. When I first started in the field, towing the net, going through this sample in the microscope, under microscope. So we know the limitations. This is a new recent review paper, just the imagination how in the future we study zooplankton. If you look at it, all those red arrows point to the imaging systems. So naturally, imaging system becomes a dominant part, potential tools for the future. So the system we built called Plankton Scope is in situ. It's a shadowgraph imaging system, self-contained, very battery powered, very compact system. So we spent a lot of efforts to design the system to make it work under a wide range of environments. So right now it can sample in the coastal water up to like with turbidity up to like 30 NTUs or even higher. So I'm not going to spend too much time on the technical details. So of course, for any imaging systems, we have the ability to take a lot of images, but can we process it effectively? That's a bottleneck. I mean, we spent a lot of time trying to figure it out. Not until like this past August, I was able to test our rapid detection algorithms to do near real-time sampling on the boat. So this is where the fast detection algorithms right now is real-time recording. So it worked pretty well. The detection speed, can be up to like 100 images per second. I mean, like our older version, like the one I just published in Eleanor methods, that's the methods I developed 3 years ago, just got out. But that one is like it can only process 20 images per minute, 20 to 40 images per minute. So this is really give us a ability to, do real-time or rapid plankton assessment. So along with those efforts enumerating copepods, zooplankton in the images, we also developed algorithms to measure the size, because size is something we care. In the Bering Sea, the main hypothesis, as I mentioned, is a switch from large copepods to small copepods, leading to the changes in recruitment for fisheries. So again, I'm not going to go into the details. The advantage of this method allows you to measure the length for instance, like the shrimp, when they fold. Traditional methods doesn't work, but this method, like oikopleura, you can actually use multiple points to detect, to measure the length. Sorry, my cats just come down and say hi to me. This is showing how it's doing the measurement. On the left side is regional images picked from the full frame images. Then we compare to the manual measurement in image J. So you can see the difference is minimum. So, okay, now I'm move on to the results. Hopefully keep everyone entertained. The first part, I'm going to talk about some results from 2017 and 2018. So focus on the yellow dots. And the first group I'm focusing on is jellyfish. So we did a lot of sampling on those two cruises. We have stationary sampling, CTD profiles, 1 meter plankton net, 20 centimeter bongo nets. We'll also have imaging system plankton scope and RBR CTD attached to the plankton scope. We'll also deploy adaptive imaging sonar systems that's sampling jellyfish because they are too big for the optical imaging systems. Then we have multi-frequency data. We have shipboard ADCP data. But gathering the data is one thing. Processing the data is another thing. Right now, for plankton scope images, we collected over close to 5 million images. As I said, before this year, we literally have no way to process the images in a short time. So we are still trying to get those data, analyzed to get more information. So for the sonar imaging systems, you can see jellyfish pretty clear. So sometimes you see the full body. Sometimes you see the major tentacles part. This is actually, you can see, is passing through a swarm of either a school of larva fish or a swarm of something, we couldn't tell, but definitely there's something there. So once the advantage for the imaging data, sonar imaging data for the jellyfish, because they sample, like, I fixed the detection range to 15 meters. In other words, if you think about it, you have it's like a pyramid going out, project out. Like you towing this triangle from the bottom to the surface, you get very fine scale resolution for vertical profiles. So those figures, this slide is probably the most difficult slides. I will spend a little bit of time here. On the left-hand side, those showing the temperature profiles in the spring, the upper panel, this one, and lower panel is summertime, late summer, late spring. So that's one. Then in each panel, the upper one showing the transects near the Alaska Peninsula here. This one corresponding here. And then the lower plots showing the transect away from the Alaska Peninsula. The color bar represent temperature, like the size of the black dots indicates the abundance for jellyfish. The right part, the right figure showing the horizontal distribution of the jellyfish, that's relatively easy, relatively straightforward. But coming back here, this figure, let's look at the springtime. The first red arrow you can see in the transect, upper transect, like close to the south. So you can see a pocket of cold water sits on the shelf. And you can also see the deep cold water off shelf. And in the northern transect, you can see the same thing. You see the cold water and getting on shelf. So in other words, this is our hypothesis. This is just a hypothesis. Our hypothesis is this is actually upwelled water being brought on shelf through tidal cycle. So you can, that's why it becomes like an isolated pocket of water sit on shelf. Those two are not connected to each other. So the phenomena is getting weaker during in the fall, right? If you look at the fall, late summer transect, I should say late summer transect, you can see that there in this transect there aren't many. There is no cold water like upwelled water present, but in the northern transect, you can still see the impact of upwelled water. Okay, now actually if we look at the distribution of the jellyfish, right? If we match this with this, it starts to make sense. This is an area where the upwelled water being constrained, then turned to the north. So in other words, close to be a convergent zone. So that's the first mechanisms the mechanism we think potentially whatever production that occurred in the canyons could be brought on the shelf. The next example is very straightforward. Then this August, we were able to sample like the areas in the canyon head labeled 1, then a transect inside of the canyon, then a transect on the shelf without the impact of canyon. So we did like 3 5-hour tows continuously up and down. Of course, our hypothesis is like because canyon heads we see upwellings, naturally we would expect to see higher krill production, more like large copepods. Then the inside of the canyons, we probably at the intermediate level shelf where there's no impact of canyons, we assume there will be very few like krill and large copepods. So this is like my collaborator Dale Kimmel from Alaska Fisheries Science Center driving the winch. So okay, again, like we collected the samples during the night. Bering Sea is we don't get dark until in September. We don't get dark until like 10:30 or 11. So the nighttime is very short. So that's we got each toes. We did nighttime sample. Then during the daytime we process images. So this is a procedure. Again, using the fast detection procedure, we can enumerate all different taxa. Then we measure the size, recording the size information. So let's look at the first transect. I'm not going to spend the time going through all the transect. The signal is very clear. So you see warmer water near bottom, cooler water in the upper layer. So yes, it's typically like upwelling signals there. So we can overlay copepods abundance. Of course, we get a lot of copepods there, almost everywhere. The difference is the size. So if we look, we start to look at krill abundance in terms of different layers. If you look at it mostly in the upper 50 meters during the night, and then from 50 to 200 meters, there are presence, but the surface, the upper 50 meters has the most abundant. And also, if you look at the presence over time, R1 is the middle night hour, so you can see the decline of the abundance as we approach dawn. So now this is the core information from this survey. The red color indicates canyon head. I labeled it as 1. Green color indicates canyon west. That's the line inside of the canyons. And then the shelf is the blue one. The upper panel showing the abundance for krill. This is as you can see, the red one, the canyon has a lot of more krills than the shelf. Shelf, we are talking about over the 5 hours in canyon heads, we encountered 870 krills. On the shelf, 5-hour toes, we encountered 13. So the difference is there. I think we can do the calculations. But copepods is a different phenomenon. So both shelf and they have a lot of copepods, but appear to be like canyon's head has the most copepods too. If you look at the size structure here, the canyons, one thing you can see, the size structure, canyon has a lot of, canyon east has a lot of large copepods, right? Then canyon west has some, and on the shelf, there are barely anything larger than 1.5 milliliter. So that's, goes back to our original hypothesis. Canyon's head really has a lots of like potential pre-items for upper trophic levels. So in summary, I make it a little bit like easier for us to understand. From the canyon inside to canyon's head, what we see large copepods and lots of krill. In the shelf, on the shelf transect, this is what we see, mostly small copepods. So goes back to the question. I think it back more why can the canyon areas serve as a refuge for the upper trophic level. The take-home message is very straightforward, and one thing I think my research is slightly different. I spend a lot of time working from seeing those invisible and move those things to ecological insights. But with the neural technology, really, I think it may make those things could happen. So I guess the next one I will show up there. So it's questions. Thanks.
Thank you. That was great. Beautiful pictures. Yeah, anybody has any questions? Oh, I see Claire. Yeah, you go.
Hello, can you see me, hear me?
Yes.
Oh,
let me see.
There we go.
I stop share.
Yeah, thank you so much. That was really, really interesting research. I was just wondering, my questions are mostly about jellyfish.
Yeah.
The imaging sonar looked amazing. Did you see, what sort of species did you see with it?
That's a good question. There are two types, two different species of large jellyfish in the Bering Sea. Most of we saw from the validation of net sampling, they are probably over 98% are Chrysioria melanoster. So, okay. Yeah.
Yeah, so quite the big ones, quite big actually, aren't they? Yeah. Did you see any of them on the acoustics? Like you had the EK, was it an EK60 you had on the? Did you see any sort of evidence of them on the acoustics? I know you didn't sample for fish, so it's quite hard to separate them, but yeah. Just out of interest of you.
If I don't know it's a jellyfish,
yeah,
then it's hard to tell.
Yeah.
If I know it's a jellyfish, then I can tell.
Okay.
Right, because a lot of the stuff I already know a jellyfish occurred in this station. When I processed the data, I sort of already have some prior information. Oh, pay more attention to something looks like a jellyfish. So if I don't have, I would say if I don't have the prior information, probably I will not pick up a lot of them. I wouldn't say all of them. I will miss a lot of them.
Yep. Yeah, fair enough. Yep. My final question, sorry, is how deep did you put your imaging sonar? I think did you put it down to 100 meters off the shelf? But
the imaging sonar, I put it down to 300 meters.
Oh, it did, yeah. Well,
yeah. So
and was it still zero for the jellies at that point, or?
Yeah.
Yeah. So are they in the canyons or not then, do you think, or was it a different place or something?
They are all out of the canyons area.
Okay.
But what happened is originally we thought they are going to be in the areas closer to the shelf where we have lots of hard substrate. But towards the end, we found we couldn't understand. Yeah, we could, because of the convergence zone, that's where they aggregated. But they are definitely not in the most. They were not most abundant in the source area.
Okay.
Yeah.
Thank you.
Welcome.
Awesome. Do you want to maybe unshare your screen so you can see people a bit better?
How did I do that?
That's a good question. Again, oh, here we go. Nice. Here we go. Chloe.
Yeah, thank you for the talk. It was interesting. I'm more a bacteria person, so it's more two curiosity question that I have. The first one is, do you think at some point with your imaging device we could imagine trying to estimate fluxes of biomass or of carbon at some point?
That's a good question, but you know, imaging technology is I would say is under a lot of tools is still under developing at this point. Just from my group, we are trying to put together like a multi-mode imaging system, which you'd utilize fluorescence imaging techniques. Then, you know, then the shadow techniques combining those two. And on the sensor end, traditionally, if we can reach a resolution to 10 micrometers, we think is pretty good. But nowadays, on the sensor end, we can go to down 1 micrometer, right? So I wouldn't be surprised in the next decade or so, there will be tools allows us to look in things much more smaller than what we can do at this point.
Okay, cool.
Yeah, I know it's not the messages you want to hear, but you know, moving those technology that in the lab to the ocean, it took a lot of efforts. It took times. So yeah.